So today, I'm gonna get started. So today we'll be talking about Campbell chapters 11 through 13. So first, Campbell chapter 11 talks about cell communication. So that's what we're gonna get started with. So there are four main types of signaling that cells use. So one of them is so three of them are local signaling, and then one of them is long distance. So the first type of local signaling is paracrine signaling, which only acts on nearby cells. So for example, certain cells can promote other cells' growth by releasing growth factors, or there are certain pro-apoptotic proteins that can promote other cells' death. And so this is also why cell medium, which is the thing you grow your cells in, that has supported old cells, can also help with it can help with new cell growth because of the growth factors that are already present in the media. So the next type is synaptic, which is when nerve cells release neurotransmitters to the postsynaptic nerve cell after an impulse. So specifically, the calcium ion influx from the outside of the cell causes neurotransmitter release through the vesicles, which binds to receptor sites, and then those pass down the action potential. And then so you don't have to worry too much about that right now because you will learn that later anyway. And the third type of local signaling is autocrine, where the signaling cell basically releases factors that acts on itself. So long distance signaling is
Wait,
called
Angela,
endocrine
your
signaling.
screen isn't
Yeah.
sharing. Your
Huh?
slides aren't changing.
I'm not changing anything.
Oh, you're not? Oh, okay.
Yeah, I'm not changing. It's all on it.
Oh, wait, it
So
just changed. Oh, wait, whatever.
what? Okay.
I mean, it was on the title slide the
Oh,
entire time.
okay. Okay. Anyways, long distance signaling is basically endocrine signaling. So these signals travel through the body to act on faraway cells. This is mostly done by hormones that flow through bodily fluids such as blood through the circulatory system. And plant cells can also do long distance signaling where the signals travel through plant vessels or diffuse through the air as a gas, for example, ethylene. And the response of the cell all depends on if it has the receptors for the signal. These receptor sites are very specific, so if the correct signaling molecule isn't detected, there's not going to be an accurate cell response. Okay, here are the three basic stages of cell signaling. So first is reception, where basically the signal reaches the receptor and as a ligand and then binds to the receptor protein on the cell. So remember the receptor is very ligand specific, so the whole transduction process here will depend on what the ligand looks like and how well it fits into the receptor. So the binding of the signaling molecule here then changes the shape of the receptor protein, and then this initiates transduction. So during transduction, the signal is essentially converted into a response down the signal transduction pathway. So this type of transduction can occur in just one step, but usually happens in a signal transduction pathway where many signaling proteins will change their shape to pass the signal down. So for example, here, there are three different transduction proteins. The transduced signal through transduction will trigger an intracellular response. So this response can be any type of cell activity, which includes catalysis. So basically, for example, you can there can be glycolysis, which is breaking down of glucose in the cell. Rearrangement of cell parts, or like the cytoskeleton, or activation of certain genes that lead to altered cell expression on in the cell, or for proteins that are attached outside of the cell. So basically, signal transduction ensures that the right activities occur at the right time and no energy is wasted. So next, so I'm going to be talking a little bit more about reception. So during reception, the signaling molecule basically binds specifically as a ligand to the receptor protein. This is sort of similar to enzymes. So there are three main types of receptors in the plasma membrane: the G protein coupled receptors, which are in the middle. the receptor tyrosine kinases, which are on the right side here. And then the ion channel receptors, which are shown on the left. So the first type of receptor is the G protein coupled receptor. So basically they're initially bound to an inactive G protein here. So however, when the receptor is activated by a ligand, the signaling molecule binding here, the shape change of the receptor basically causes the inactive G protein to activate through GTP, and then this GTP becomes GDP, and basically moves it to the next protein for it to carry out its functions. So as you can see here, the G protein is moving onto the next enzyme here. So this activated G protein, after dissociation, can change the shape of the next enzyme, which then leads to further cellular processes. Mutations in this pathway can be very harmful and lead to cancer, since if the G protein is always active, the cell will continue this pathway even without stimuli, and then proliferate or produce its proteins or these cellular responses excessively. Okay, next are receptor tyrosine kinases. So they belong to the protein kinase group, which basically transfers phosphate groups from ATP to another protein. Specifically, tyrosine phosphate kinases attach phosphates to tyrosine, which is a TYR here. So initially, these receptor tyrosine kinases are two separate units, as you can see here. But then once the signaling molecule attaches to the receptors, they come together to form a dimer, which is a bigger unit of two little smaller units. This dimer is then capable of transferring phosphate groups from the ATP, which is seen here, and the phosphate groups are attached onto the tyrosine, and then these can be passed on to other proteins to continue the message. So next are ion channel receptors. So an ion channel receptor is usually closed to its specific ion type. So it's usually closed like this, so nothing can flow through. However, when the ligand binds to the receptor, the ion channel is open here, and then it allows the ions to flow through. So when the ligand stops binding to the receptor, the channel then closes back up and the ions stop flowing. And this is very important in cells such as neurons that depend on ion channels to control neural activity. Okay, in addition to extracellular receptors, there are also intracellular receptors. So these receptor proteins are inside the cell as opposed to embedded within the membrane. So how do the signals get to them? Well, the signals are usually hydrophobic, such as hormones like aldosterone, as shown here, or also extremely small, so they can pass through the membrane. These signaling molecules, since they are hydrophobic or very tiny, can pass through the nonpolar plasma membrane directly into the cell without a protein to relay the message. So there, the signaling molecule will bind to an intracellular receptor here to cause an intracellular response. And these usually involve the signaling molecule traveling into the nucleus and affecting protein expression. So the signaling can also often affect transcription factors that aid in protein transcription and indirectly affect protein translation and production. So examples of these intracellular ligands include steroids, which are lipids that are very nonpolar, so they can flow easily through the cell membrane. And they also include certain gases such as nitric oxide, which are also very small and also relatively nonpolar, so they can just go directly through the cell membrane. Although transduction can have single steps, multi-step transduction pathways allow for more coordination and control within the cell, which helps to regulate the response. So in transduction pathways, relay molecules activate other relay molecules down the chain. So most of these relay molecules are proteins, And it is important to remember that by this time, the signaling molecule itself is not passed down, but it is the reeling molecules at work that are passing the signal down. So these signaling molecules, the first messengers, usually, unless they're intracellular receptors, they will not directly go into the cell, but it is the proteins changing shape that helps pass the signal down. So phosphorylation, which is the addition of a phosphate group, is really important in signal transduction. So the phosphorylation is carried out by protein kinases, as we have said before, and that basically involves adding a phosphate group. So phosphorylation causes shape changes in the protein, since an extra group is added, and then the shape change results in a phosphorylation cascade, where the initial phosphorylated protein basically transfers its phosphate group onto the next protein, which then does the same to the following proteins. So protein phosphatases are also very important in signal transduction, since they will remove the added phosphate groups after the signaling molecule basically decreases in level. And so this ensures that the signal transduction pathway can be turned off very quickly after the ligand is no longer present, to avoid like any unnecessary or unwanted effects. So small molecules and ions that help pass the message down are called second messengers. Sorry, second messengers. The first messenger is basically the ligand that binds to the receptor, and these second messengers are often smaller and water soluble, so they can diffuse throughout the cell quickly. The two most widely used second messengers are cyclic AMP and calcium ions, and there are many proteins that are sensitive to the changes in the concentration of these second messengers within the cell. So specifically, cyclic AMP here is made by adenylyl cyclase after a G protein coupled receptor. And this is important since it carries signals for glycolysis within the cell. So similarly, calcium ion influxes in neurons activate the release of neurotransmitters to the postsynaptic neurons in vesicles. So another one that is pretty common is called IP3E. So the responses from a signaling pathway usually result in changes or regulation of gene expression. So for example, as discussed before, transduction can activate transcription factors or control their expression to promote a certain protein synthesis. Other types of signaling molecules, such as growth factors, can also help regulate the cell cycle and cell division. So the response can also be regulated in many ways. So first, the signal can be amplified with enzyme cascades. So essentially, each enzyme continuously activates more and more enzymes, resulting in a lot of proteins and enzymes being activated ultimately. So for example, if I have one signaling protein, it will activate 10 more, and then these proteins will each activate 10 more. That basically and results in a lot more proteins being activated and a lot larger response. So similarly, scaffolding proteins, which are seen on the bottom right here, can also help with amplification, since there can be many relay proteins attached to one scaffolding protein, and so all of these proteins will be affected at once. And then the response to the same signal could also vary from cell to cell depending on the type and availability of receptor proteins and transduction proteins. So for example, one cell can respond well to one ligand since it has many receptors specific for that ligand, but another cell type may not respond that well to the ligand since it doesn't have the correct receptor proteins. Okay, the binding of signaling molecules to the outside receptors is reversible. And so when the external concentration of signaling molecules falls, the binding of these proteins to receptors is also reduced. When the signal binding falls below a certain threshold, the cellular response is then terminated. So the receptor returns to its inactive form, and the protein phosphatases remove any remaining phosphates, and the proteins return to their inactive form. So this basically prevents excessive responses to a certain signal after the signals are no longer very present. Sometimes multiple cell signaling pathways are integrated. So an example of this is seen in apoptosis, which is controlled cell suicide. So the signal for apoptosis can be both intra or extracellular signals, such as the tumor necrosis factor. When cells receive these factors, the DNA and intracellular materials are basically cut up, digested, and then the cell form blebs and start to die. So, sorry, although apoptosis might seem bad, it is actually good for the development of certain body parts, such as fingers. So when you're a baby, the tissue behind your fingers, like these parts, undergo apoptosis, and that's how you get your individual fingers instead of like big fleshy things as your hands. Okay, that is chapter 11. So we'll be going on to chapter 12, which is talking about the cell cycle and mitosis. Okay. Wait, hold on. Do we have any questions? Oh. Yeah, okay. I'm gonna continue then. So next, we're going to be talking about the cell cycle, which is mitosis. Oh, my bad. So first, a cell's DNA, which is their genetic information, is called its genome. So the length of DNA is very, very long, and so this DNA must be efficiently packed into cells. This DNA is packed into chromosomes, which is made up of chromatin. This chromatin is composed of DNA wrapped around histone proteins, and then this chromatin is packaged further into the chromosomes, as you can see here. So during mitosis, a single chromatid basically, like before mitosis actually, the single chromatid replicates into two sister chromatids, which are attached at the centromere in the middle. So each one of these is a separate sister chromatid. And these chromatids have telomere ends that basically protect the DNA from degradation and damage. So during mitosis, these two sister chromatids are pulled apart and then reproportioned into the two resulting daughter cells, as we will discuss later. So under normal conditions, there are 46 chromosomes in your somatic cells, which are your body cells. And so we notate this as 2n. Half of these chromosomes will come from your mother and half from your father. In gametes or sex cells, there are 23 chromosomes, so which we notate as n. So when you have a kid, you pass these chromosomes from your sex cells onto your kid cells, which then ends up with 46 chromosomes because of the other parent. So out of these 23 chromosomes, one of them is a sex chromosome, either X or Y. So if you have two X chromosomes in your somatic cells, you are a female. And if you have an X and a Y chromosome, you are a male. So there can actually be certain exceptions to this because of some, I guess, like disorders or issues that come up, but this is basically what generally happens. So mitosis, which we will be talking about, is the separation of these chromosomes in the cells. And then cytokinesis, which is part of mitosis but is a little different, is the division of cellular content that follows mitosis. So the cell cycle includes mitosis as well as the time period between divisions, which is called the interphase. So during interphase, the cell mainly grows and develops, and the subphases in interphase are divided into G phases, which are cell growth, and the S phase, which is synthesis, so like basically DNA replication, and that's when all the replication happens. So during the G1 phase, the cell experiences the fastest cell growth. And so the cell basically just obtains its nutrients and carries it out its normal cell activities. And then it moves on to the S phase, where the DNA is replicated so that enough DNA can be portioned into each daughter cell after replication. So basically the single chromatid, which we saw before, Well, during this time, it's not condensed into a chromosome yet, but basically its material will be copied so it has enough for another sister chromatid. Okay, and then the cell basically proceeds into the G2 phase where the cell grows even more, but with double the amount of genetic material. So during interphase, there can be many checkpoints. So after the G1 phase, there is a checkpoint in which certain cells might move to a phase called G0, where no replication happens and the cell basically just lives. We'll be talking more about this later as well. So after interphase comes the mitotic or M phase, where the cellular content is divided along with genetic material. So here is a little diagram of what happens in the cell cycle. So most of this is interphase. And then there's mitosis and cytokinesis, and then these cells repeat the whole cycle again. So mitosis also has its own subphases. So the first phase is prophase. And during this phase, the chromatin, which usually you can't see under the microscope, it will condense and become visible under a microscope. So these become arranged in sister chromatids, and then those become arranged in chromosomes. So the nucleoli, which is in the center of the nucleus, also disappears. And then also the mitotic spindle begins to form and assemble from centrosomes, which and this mitotic spindle consists of the centrosome and its microtubules that are growing out. So here is some early mitotic spindle that is forming. So the second phase is called prometaphase, which is between prophase and metaphase. So during this time, the nuclear envelope fragments and disappears. And also the microtubules start to invade the nuclear area. And so on the sister chromatids, little, I guess, basically hooks like called kinetochores, they form at the centromeres. And so these microtubules here will attach to these kinetochores and start to move the sister chromatids around. Okay, next is metaphase. So in metaphase, the chromosomes are basically pulled by the microtubules into the middle of the cell, which is called the metaphase plate. The kinetochore microtubules are now all attached to the sister chromatids on the kinetochore. And then comes anaphase, where the cohesin proteins, which hold the sister chromatids together at the centromere, are cleaved, and then the sister chromatids are pulled apart by microtubules. So while the non-kinetochore microtubules are not attached to the chromatids elongate, the kinetochore microtubules that are attached to the chromatids shorten, and then they pull the chromatids to the opposite poles of the cell. And in doing this, the cell also elongates. And then during telophase, the two new cells basically form, but in one big cell. So the nuclei and the nucleoli reappear, the nuclear membrane regenerates, and the chromosomes uncondense. So everything's moved to the two sides now. And then lastly, in cytokinesis, the cellular content is sorted and the cells are physically divided, as you can see here. So during cell division, cytokinesis occurs by cleavage, where a cleavage furrow forms to split the two cells. So the cells are basically cleaving themselves in to separate. In this process, actin and myosin microfilaments on the cytoplasmic sides of the cells inside here work together to contract, which causes constriction at the center of the cell. And then the cells will separate themselves. So this process is a little different for plant cells because plant cells have a very hard outer cell wall and that is unable to form a cleavage furrow. So in plant cells, the vesicles in the cell with cell wall material will aggregate material in the middle of the cell to form a cell plate from the inside out. So this cell plate is made of cell wall material and then will continue to grow until it fully divides the two cells and forms the new cell wall. Okay, bacteria, on the other hand, undergo binary fission. So this basically just involves replication of genetic material followed by cytokinesis of the cell. So the genetic material, the nucleoid, is basically replicated and then it is split apart, and the bacteria pinch in and then separate. Okay. To ensure that the cell cycle doesn't go crazy and to keep the cells replicating for too long and cause cancer, the cell cycle regulators help keep the cells in check. So one type of regulating molecule is the cyclin-dependent kinase. So to function, these kinases must be attached to cyclin. These cyclin-dependent kinases are always present but are only activated by cyclin. So an example of a cyclin-dependent kinase is the maturation promoting factor, or MPF. So this MPF can basically be thought of as the M phase promoting factor, since it allows cells to proceed into the mitotic phase after interphase. So during interphase and as the cell finishes interphase, there are also cell cycle checkpoints that can stop the cell if there is excessive damage to the DNA or the cell. Between the G1 and S phase, as we have talked about before, there is a cell cycle checkpoint that can move a cell to the G0 or non-dividing phase. So actually, a lot of neurons are in the G0 phase and they don't replicate themselves. So this does not only have to be for DNA or cell damage. Also, before M phase, there's another checkpoint to ensure the DNA that is replicated already is not damaged, and then the cell can proceed. Furthermore, signaling molecules outside of the cell, like growth factors, can promote cell growth and division. However, when the cells grow too clustered, there's going to be density-dependent inhibition, which limits the cell growth to prevent excessive competition, and so the excess cells will start to die. Okay, so failures of regulation during the cell cycle can cause cancer, which is uncontrolled cell division. So basically, when these processes basically fail, the cells can start dividing uncontrollably, and that is basically cancer. So there are two types of tumors that can form. The first type is benign tumors, and these tumors are local and don't metastasize or move to new areas. So this can, well, all cancer is bad, but benign cancers are not as bad as malignant cancers, since these cancers will not move around and they're local, so you can remove them, often with radiation or surgery. On the other hand, malignant tumors are very bad because they can spread to new tissues and are very serious, and then these can actually come up after months or years after initial treatment. Okay, that is all I have for chapter 12. So we can take about 5 minutes and then come back to chapter 13. Okay. I think we can go and continue. Oh, there hasn't been 5 minutes though. Hold on, give me a second. Okay. So next we'll be talking about meiosis, which is a little bit similar to mitosis, but has a completely different function. So, during inheritance for offspring, the genes of the baby are always inherited through gametes, which are sex cells that are egg and sperm, and never somatic or body cells. So there are two types of reproduction. In asexual reproduction, the organism makes genetically identical copies of itself. So this is seen in bacteria, where the bacteria just undergoes binary fission and becomes two organisms that are genetically identical to the one parent organism. Meanwhile, in sexual reproduction, the gametes of two parents are always combined to create genetically unique offspring. Oh, sorry, that wasn't 5 minutes. Well, we'll have a lot to get through. So, okay. Yeah. Okay, so in each human body cell, as we have talked about before, there are 46 chromosomes, which include two sex chromosomes. However, in gametes, there are only 23 chromosomes, including one sex chromosome. So basically, two gametes must combine to form an organism. So when the two chromosomes from the two gametes combine in a new organism, the resulting chromosome pair is called a homologous chromosome. So as you can see here, there are actually 4 sister chromatids in total. So these two sister chromatids on the right are from one parent, and then the other two originate from another parent. So these homologous chromosomes encode for the same protein type, but can have variations in the loci or the location of the gene. So for example, the loci of eye color is at the same spot on homologous chromosomes, but one can code for green eyes while the other can code for brown eyes. So these homologous chromosomes we're talking about are autosomes, which are not sex chromosomes. The sex chromosomes, however, don't really follow a homologous chromosome pairing. So the combination of X and Y chromosomes determine the sex of the offspring. So two X chromosomes in the zygote lead to a biologically female offspring, while an XY pairing can lead to a biologically male offspring. So we'll talk about this a little more later, but the sex chromosomes basically, if you have two X chromosomes, you can only pass on X chromosomes. However, if you are a male and you have an XY pairing, you can either pass on your X chromosome or your Y chromosome in your gametes. So the number of chromosomes in each cell basically determines the type of cell it is. So haploid cells only have one set of chromosomes, and gametes are basically a great example of this, as they only have one set of the 23 chromosomes. Two haploid gametes will combine into a diploid cell during fertilization, and diploid cells, on the other hand, have two sets of chromosomes, which are the body's somatic cells. So here's a little, I guess, cycle of fertilization and meiosis. So during meiosis, the diploid cell basically makes itself into haploid cells, which then combine. So two haploid cells combine into a diploid cell, and then the cycle will repeat. During meiosis, a diploid parent cell is divided up into haploid cells. So meiosis is somewhat similar to mitosis, except the phases will repeat 2 times. So there is meiosis 1 and meiosis 2. So all of both meiosis 1 and meiosis 2 have prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. However, they're basically just divided into 1 and 2, and the genetic material gets divided up a little bit differently than before. Also, the daughter cells that come out of this process are haploid with only one set of chromosomes instead of being diploid. During meiosis 1, the homologous chromosomes, which replicate beforehand, so this is your 2N cell, and then it replicates into so these are your homologous chromosomes. There are 2N, and each will replicate into having its own sister chromatid. So before the separation, there is crossing over between the homologous chromosomes as they line up in the middle of the cell. So essentially, the genetic material between the loci of 2 homologous chromosomes are swapped. And so this helps create more variation in the produced gametes. So here in meiosis 1, basically the replicated homologous chromosomes are separated into 2 daughter cells. And then during meiosis 2, the sister chromatid of each chromosome of each replicated chromosome will be split up into more daughter cells. And so each produces a gamete, which is left with only one set of chromosomes. So while in mitosis, you get a result of 2 daughter cells in total from one cell. During meiosis, you get 4 daughter cells from one cell, and also during mitosis, you have diploid daughter cells, but in meiosis, you get haploid daughter cells. So meiosis will help to create genetic diversity in many ways. So the first way is through independent assortment. So during meiosis, when the homologous chromosomes line up, they can line up in random assortments. So this is your possibility 1. So basically the blue chromosomes are on one side, and then the red are on the other side, and then they line up to split apart very nicely. And then your sister chromatids also line up very nicely to form gametes like such. However, there is another possibility where your blue and red chromosomes don't line up in the same orientation, and then this can give you different arrangements of sister chromatids in your daughter cells, as you can see here. So this allows for random assortment of these chromosomes into the daughter cells, and the same goes for meiosis 2. So here, where the sister chromatids are assorted into gametes randomly. So the second way is crossing over, which is seen during meiosis 1. So as we have talked about here, there's crossing over when the chromatids line up like this. Sorry, not when the chromosomes line up like this. So during crossing over, the genetic material between the homologous chromosomes are exchanged, and then this creates recombinants that have slightly different genetic combinations, which are passed on to offspring. So your gametes are not going to be genetically identical because of this, and then this can create a variation that can be passed on to offspring. So lastly, random fertilization, which is basically random sperm fertilizing with unique genetic material fertilizing random eggs, ensures the fertilization is by chance. Okay, so here is a little picture of crossing over. So when the homologous chromosomes are lined up, their genetic material can be swapped like this, and then when these sister chromatids are passed on, each will be a little bit different from before, and so that creates your variation. Okay, we're done with chapter 13. I'll stop sharing for a bit, but we have some questions we can go through. Hold on. Okay. I'll screen share some questions that are off of Usable. So wait, what? So you want the sperm race? Well, the sperm do like survive longer than the other one, so I guess you are special, maybe. Um, yeah, but I mean, you have like millions of sperm, and only like one of them, which is pretty random, gets to fertilize the egg. So yeah, you're all special. Okay. So this one is like a two-part-ish question, but number three is not very, so the which enzyme does this drug inhibit is not very related. So okay, I don't know how to do a poll, but okay. So this thing is a skin disorder that is caused by an increased proliferation of skin cells, blah blah. Skin cells from a mouse model for this disease are isolated and cultured in a plate containing a drug that promotes the supercoiling of DNA. So which enzyme does this drug inhibit? This is not really related, but which phase of the cell cycle do cells treated with the drug specified in question 3 become arrested? Answer please. Oh, yeah, these are open questions. Or why are only like 10 people answering these? Imagine. Okay. Um, oh, okay, so a lot of you guys put B or D, which is not the correct answer. The correct answer is E. I don't know why you would think it's B or D, but so during S phase, basically your DNA gets replicated. So when the DNA gets supercoiled, it's not very replicated. During G0 phase, the cell isn't really doing anything. It's just like living. During G1 and G2 phase, the cell is just like it's growing. So it's growing phases, and during M phase, it's basically where it replicates. So the DNA part has the most to do with S phase. Okay. Okay, next. So a replication inhibitor is added to stem cells in the middle of DNA replication. At what phase of the cell cycle would these cells remain? Derek, what is going on? Okay. Um, Voicing people, that's pretty much better. Okay. So most of you put D, which is good because that is the S phase where the DNA replicates and is like synthesized. So yeah, good job. Oh, and A is not correct because that's like this like the actual division of this cell. Okay. Which of the following is false about meiosis 2? Cytoplasmic division occurs, individual chromosomes line up on the equatorial plate. Oh, by the way, the metaphase plate is also called the equatorial plate. Sister chromatids separate and move toward opposite poles. The chromosomes replicate and the nuclear envelope disintegrates. Okay. So most of you answered D. Good job. Um, so during meiosis 2, the cells are split apart because that's what happens. Meiosis 2 includes metaphase 2, where the individual chromosomes line up on the equatorial plate. The sister chromatids also move towards opposite poles, which is anaphase 2, and the nuclear envelope also disintegrates, which is prometaphase 2. However, the chromosomes don't replicate because that happens before meiosis starts. So yeah, that's why it's false. Okay. Other than that, I have a couple more questions that are not from the open, but it's like general knowledge and good to know. Okay, so I would like got the second one wrong. Oh, too bad. Okay, so I'll type the answers in chat and I would like you guys to like message me the answer. Okay, so first, what are some differences between mitosis and meiosis? I would like you to tell me two differences between mitosis and meiosis. Yeah, good answers. Okay. And when they're half the parent cell number, it's called haploid. It's a good term to know. So there is not uneven gene distribution, I would say. There is just crossing over, so it creates more variation, but the genes are still distributed evenly in normal circumstances. Yeah. So mitosis does have an output, I guess, of 2 cells. Mitosis cells don't have 46 haploids, they have 46 chromosomes. So the chromosomes are the actual genetic information things, and then the haploid and diploid are the terms for how many chromosomes they have, chromosome pairs. Okay. So what is the secondary messenger in a G protein coupled receptor pathway? Okay, hold on. Give me a second. So mitosis isn't for prokaryotes. Prokaryotes, they undergo binary fission, which is just regular division of. So it's just the splitting of the cells after DNA replication. And then meiosis is not the exchange of information, it involves the exchange of information, which is crossing over between chromosomes. Okay. Guys, I'd like you to guess if you don't know. Derek, bro, what?
Answer the question. Answer the question.
What's the question? Second messenger in the G protein coupled receptor pathway. Help me out here. Help me out here. I do physics, bro. I do physics. Help me out here.
Cyclic AMP.
What?
Cyclic
Cyclic AMP. What is it? Cyclic AMP?
AMP.
AMP.
Yeah, it is cyclic AMP. So
Oh,
Oh.
physics on top.
So GTP isn't the second messenger, it's what provides the energy for G proteins to like go to the adenylyl cyclase to produce the cyclic AMP. Yeah, okay. We're good. Yes, it is the camp. Don't zone off, guys. You need to know this. I mean, yeah, okay. We're at 8 p.m. So thank you for coming, everybody. We'll see you next week.